nonpolar. Our final two functional groups are the phosphate and the sulfhydryl group. The phosphate group is very much like the carboxyl group in that it carries a negative charge when it's inside of a cell. It liberates hydrogen ions, so it is acidic, it is polar, it is hydrophilic. It is also unique from the carboxyl group in that it has the ability to form bonds that contain a lot of energy. So it's considered to be a high energy molecule. That's why it's part of our energy carrying molecule ATP or adenosine triphosphate. Its location can be found in nucleotides or nucleic acids, such as DNA and RNA, phospholipids, and proteins. The final functional group, sulfhydryl, is a group that is prevalent in amino acids and proteins. And this group is a sulfur with a hydrogen bound to it through a single polar covalent bond. But in most cases, when you find it in a larger molecule, it forms what are called disulfide bridges, which means two sulfur groups bind together. And since they're bound together and they're the same molecule, they are bound through a non-polar covalent bond. These sulfhydryl groups and disulfide bridges are common in proteins, and proteins are built upon amino acids. So some amino acids will have these sulfhydryl groups. So what do cells do with organic compounds? Well, in essence, cells are built upon organic compounds through condensation reactions, where two smaller molecules will covalently bond to become one larger molecule. So that's how when you take amino acids that your body has absorbed from your digestive system and your body puts them into a new protein, that's the reaction that's occurring. That's a condensation reaction. A hydrolysis reaction is the reverse. So think about when you eat food for fuel, but you also eat it for raw materials. So if you eat protein, which is made up of amino acids, you're going to take the protein, break it down into amino acids, and then those individual amino acids will be strung together into new proteins that your body needs. The functional groups are the groups that help characterize the features that the macromolecules have. So they're common throughout the macromolecules, and they act the same no matter what macromolecule they're located in. Depending upon how the organic molecules are put together or rearranged, you can convert one organic compound into another. And you can also, with those organic compounds, move electrons. And when you move those electrons, you in essence move energy. So let's step back and look at condensation and hydrolysis. These are the foundational reactions for macromolecules. Condensation builds macromolecules, hydrolysis breaks them down. So in this example, we have two carbon-based molecules. We have the one to the left with a 6-carbon ring and an OH, and the one to the right with a 6-carbon ring and an OH. You can see that we're going to add them together, represented by the plus sign. The arrow shows what they make. And notice what they make are the two 6-carbon rings now bound through an oxygen. But we had to lose the whole OH from the molecule to the right and the hydrogen from the molecule to the left. So we've created or made water from the OH of the molecule to the right and the H from the molecule to the left. This is a condensation reaction. We've taken two simple molecules, two 6-carbon rings, and put them together into one larger molecule. And as we did that, we had to free electrons so that they can bind. And when we freed, the way that we freed electrons was to remove a molecule of water. So the simple 6-carbon molecule to the left and the simple 6-carbon molecule to the right have now been bound together by electrons that were liberated by removing a molecule of water. For this reaction to occur, you do need help, and that comes in the form of an enzyme. And an enzyme is a biological catalyst, which means that it exists in cells to expedite or make chemical reactions more feasible for the cell. The opposite of condensation is hydrolysis, and this is where we take the more complex molecule, like the one that we just built, and you can see that at the top of the slide, and we break that more complex molecule into its simple units. We do that with the aid of an enzyme and by adding water. So we basically are just reversing a condensation reaction. When we reverse this condensation reaction, we add the parts or the components of the water molecule back to the individual units. So that now our two individual units, the 6-carbon molecule on the left and the 6-carbon molecule on the right, are stable. In essence, condensation is building up. That is a branch of metabolism called anabolism, and hydrolysis is breaking down. That is a branch of metabolism called catabolism or digestion. So now we're going to get into the 4 macromolecules. And the 4 macromolecules are built upon individual atoms which come together to form molecules, which then come together to form macromolecules. And when I say come together, what I mean is bonding. So this is a view of some gumdrops, and on the surface of the gumdrops we can see sugar. Sugar is a type of carbohydrate. So let's look at our first macromolecule or carbohydrate. Carbohydrates are built upon sugar units. Here we have 2 sugars represented, glucose to the left and fructose to the right. Notice how I've boxed in the hydroxyl on the fructose and the hydrogen of a hydroxyl group on the glucose. This is going to be where we form our covalent bond between the glucose and the fructose, making sucrose. And of course, we have liberated water. So if you remember, this is a condensation reaction. So glucose and fructose are monosaccharides under the category of carbohydrates, and sucrose is called a disaccharide because it's made up of 2, di meaning 2, saccharide meaning sugar. So sucrose is a disaccharide made up of 2 sugars, whereas glucose and fructose each are monosaccharides, each being made up of 1 sugar. Multiple sugar units can then be strung together, forming complex carbohydrates. And we can see 3 different complex carbohydrates in this image. The one on the upper left, A, is cellulose, and this is a structural component of plants. It basically makes up the stringy nature, so to speak, of the cellulose. It's very fibrous, and it helps keep plants stable and able to stand structurally very, very tall. In B, you can see another form of a complex carbohydrate. This is called amylose, which is basically plant starch. And plant starch is an energy molecule. And we have starch in us as well. That's what C is representing. Glycogen is animal starch. And in both cases, starch is an energy reservoir. It is a way to hold energy within the bonds between the sugar units that cells can then easily access. A is not considered to be an energy reservoir because of the nature of those hydrogen bonds between those side chains in the cellulose. Enzymes can't get in there and break them apart. That's why we don't digest cellulose in our digestive system. That's considered to be our roughage. That passes out as our part of our waste from our large intestine. But B and C, we can break down. B, when we eat plant starch, think of French fries or potatoes. As we digest that material, we get the individual sugar units, and then we take those individual sugar units and build them into our own energy reservoir, which we see on the right, which is glycogen. And glycogen is stored in our liver, and when we need some sugar, like let's say between meals, our body will enzymatically take those sugar units of the glycogen in our liver and break them off, kind of like little energy packets. And then they will be used in a process called cellular respiration, which is week 3.